Fertilization. Fertilization is the process by which male and female gametes fuse to form zygote. Or fertilization means the fusion of two mature germ cells or ovum and a spermatozoon to form a single cell, the zygote. Normal size. Ampullary region of the uterine tube. Process of fertilization. Transport of spermatozoa. 200 to 300 million spermatozoa are deposited in the female genital tract. Among them, 300 to 500 reach the site of fertilization. Only one of them fertilize the egg. Others help in better penetration. Transport of ovum. The ovum transports into the uterine tube and meets the sperm in the ampullary region, the usual site of fertilization. Contact and fusion of the gametes. A direct contact between the spermatozoa and the zona pellucida of the ovum is necessary for fertilization. Spermatozoa are not able to fertilize the oocyte immediately upon arrival in female genital tract but must undergo capacitation and acrosome reaction to acquire this capability. Better barrier penetration. It includes the three phases. Penetration of corona radiata. Corona radiata. Capacitated sperm pass freely through corona cells. The corona cells are dispersed mainly by the action of the enzyme hyaluronidase. The corona are dispersed mainly by the action of enzyme hyaluronidase. Release from the acrosome of the sperm. Penetration of zona pellucida. Acrosome reaction allows the sperm to penetrate the zona pellucida. Once the sperm penetrates the zona pellucida is zona reaction. A change in properties of zona occurs. Such reactions makes the zona pellucida permeable to other sperms. A zona reaction, change in properties of zona occurs. Fusion of the oocyte and sperm cell. After adhesion, the plasma membrane fuse and break down at the era of fusion, leaving the plasma membrane behind. The head and the tail of the sperm enter the cytoplasm and the oocytes. The head and the tail of the sperm enter the head of the tail. The head and tail of the sperm enter into the cytoplasm of the oocyte. Fusion, leaving the plasma membrane behind. Completion of the second mitotic division of oocyte and formation of female pronucleus. Immediately after entry of the sperm of the oocyte finishes the second mitotic division. Immediately after entry of the sperm, the oocyte finishes second mitotic division and forms a mature ovum and second polar body. The nucleus of the mature ovum becomes vesicular and is known as female pronucleus containing haploid chromosomes. Female pronucleus containing haploid chromosomes. Formation of male pronucleus. Within the cytoplasm of the oocyte, the nucleus of the sperm becomes swollen and becomes a male pronucleus. DNA replication. Replication of the DNA occur in both pronuclei and during their growth. Breakdown of the pronucleus membrane. Condensation of the chromosomes are arranged of the chromosomes for mitotic cell division. The combination of 23 chromosomes in each pronucleus results in the zygote within 46 chromosomes. Factors upon which fertilization depends. Time of ovulation, time by which sperm releases the uterine tube, number of sperm, variability of sperm ovum, results of fertilization, restoration of diploid number of chromosomes, termination of sex and new individual, initiation of cleavage, termination of sex of the individual, restoration of the diploid number of the chromosomes. Factors upon fertilization depends. Time of ovulation, time of sperm reaches the uterine tube, number of sperm and viability of sperm ovum. What is a zygote? A single cell fertilized ovum is called a zygote. Explained anatomically and biologically. Father is responsible for the termination of sex of the baby. Father is responsible for the termination of sex of the baby. An X carrying sperm produces a female embryo and a Y carrying sperm produces a male embryo. As due to the presence of Y chromosome, the embryo becomes male. which is present in the father, not in the mother. So the father is solely responsible for the determination of the sex. What changes occur in the spermatozoa upon arrival of the female's genital tract? Changes occur in the spermatozoa upon arrival in the female genital tract. Spermatozoa are not available to fertilize the oocyte immediately upon arrival in the female genital tract. They must undergo capacitation and acrosome reaction to acquire the capability. Capacitation এবং acromial reaction, acrosome reaction, capacitation and acrosome reaction. Purpose of these changes: capacitation, it passes through the corona cells and undergo acrosome reaction. An acrosome reaction, it passes through the zona pellucida. It passes through the corona cells and undergo acrosome reaction. It passes the zona pellucida. What do you mean by capacitation? Capacitation is a period of conditioning of sperm and male and female genital tract to acquire the ability to fertilize ovum. Time approximately 7 hours. The period of condition of sperm in the female genital tract to acquire the ability to fertilize the ovum, approximately 7 hours. Process: during capacitation, a glycoprotein coat and a seminal plasma protein are removed from the plasma membrane and ovalis, the acrosome region of the spermatozoa. Only capacitated sperm can pass through the corona cells and undergo acrosome reaction. During capillary reaction, a glycoprotein coat and a seminal plasma protein are removed from the plasma membrane and overlies the acrosome reaction by spermatozoa. Only capacitated sperm can pass through the corona cells and undergo acrosome reaction. During capacitation, a glycoprotein coat and the seminal plasma proteins are removed from the plasma membrane. Define acrosome reaction. It is the reaction characterized by multiple points of fusion between the plasma membrane and acrosomal membrane of sperm that permits release of acrosomal contents to overcome the barriers around the oocyte. The acrosome reaction occurs after binding into the zona pellucida. It induced by the zona proteins. This reaction culminates the release of enzymes needed to penetrate the zona pellucida, including acrosin and trypsin-like substance. Zona pellucida, acrosin and trypsin-like substance. What do you mean by zonal reaction? Zona reaction. The zona reaction refers to an alteration in the structure of the zona pellucida, making it permeable to other sperms, catalyzed by proteinase form of cortical granules. Zona reaction. The zona reaction refers to an alternation in the structure of zona pellucida, making it impermeable to other sperms, catalyzed by the process of cortical granules. Description: the zona is a glycoprotein shell surrounding the egg that facilitates the maintenance of sperm binding and includes the acrosomal reaction. Both binding and the acrosomal reaction are mediated by the region GP3, a zona protein. Release of cytochromal enzyme allows the sperm to penetrate the zona, thereby coming in contact with the plasma membrane of the oocytes. Permeability of zona pellucida changes when the head of the sperm comes in contact with the oocyte surface. This contact results in the release of lysosomal enzymes from cortical granules lining of the plasma membrane and oocytes. In turn, these enzymes after properties of zona pellucida to prevent sperm penetration and inactivates a sperm-specific receptor site for spermatozoa on the zona surface. Distinguish between the following two terms: acrosomal reaction and zona reaction. Diffuse between acrosomal reaction and zona reaction. Acrosomal reaction. Definition: it is the reaction characterized by multiple points of fusion between the plasma membrane and the acrosomal membrane of a sperm that penetrates release of acrosomal contents to overcome the barriers around the oocytes. Zona reaction. Zona reaction refers to an alternation in the structure of zona pellucida, making it permeable to other sperms catalyzed by protease from cortical granules. Alternation of cortical granules. Importance. It allows the spermatozoa to penetrate the zona pellucida and fuses with the oocyte membrane. It blocks the sperm to penetrate the zona pellucida and thus block the polyspermy. So the first one prevents polyspermy and the second one zona reaction. Polyspermy that allows spermatozoa penetrate the zona pellucida and fuses with the oocyte membrane. Acrosomal reaction. The spermatozoa penetrates the zona pellucida and fuses with the acrosomal membrane. How is polyspermy prevented? How is polyspermy reaction blocks the sperm of penetrate zona pellucida and thus blocks polyspermy? The zona reaction blocks the sperms to penetrate the zona pellucida and thus blocks the polyspermy. The zona reaction blocks the sperms to penetrate zona pellucida and thus blocks the polyspermy. The zona reaction blocks the sperm to penetrate the zona pellucida and thus blocks the polyspermy. Mention why normal fertilization occurs at that ampullary part. Normal fertilization occurs at the ampullary part. Fertilization occurs at the ampullary part. Normalized fertilization occurs at the ampullary part of the uterine tube because this is the widest part of the tube and is the closest to the ovary. Because this is the uterine tube because it is the widest part of the tube and thus close to the ovary. Short fertilization in vitro. In vitro fertilization. In the artificial process which humans ova and embryo transfer is conducted by laboratory steps of IVF. Follicle growth in the ovary is stimulated by administration of gonadotropins. Oocytes are recovered by laparoscopy from the ovarian follicles with an aspiration just before ovulation, when the oocyte is the late stages of first meiotic division. Oocytes are recovered by the laparoscopy from ovarian follicles with an aspiration just above the ovulation, when the oocytes is at the late stage of the first amniotic division. The egg is placed in a simple culture medium. The sperm are added immediately. Fertilization eggs are monitored on the slight cells change and then placed in the uterus to develop in term. Advantage. Fortunately, because pre-implantation stage embryos are resistant to teratogenic insult, the risk of producing malformation offspring by the in vitro presence is low. Embryos are resistant to teratogenic genetic insults. Disadvantage. A disadvantage of IVF in vitro fertilization is low success rate. Only 20% of fertilization via implantations are developed to term. Therefore, to increase the chance of successful pregnancy, 4 or 5 ova are collected, fertilized, and placed in the uterus. This approach sometimes leads to multiple births. Explain from your knowledge of anatomy of genetic determination of sex occurs at fertilization. Determination of sex occurs at fertilization. During fertilization, a chromosome from the sperm cell, either X or Y, fuses with the X chromosome of the ovum. An X carrying sperm produces a female embryo and a Y carrying sperm produces a male embryo. Hence, the chromosomal sex of the embryo is determined by the time of fertilization. 1 week of development. Mention the events first of development, short note, cleavage, and blastocyte. 1 week of development. The first week of human development begins with fertilization of the egg by sperm forming the first cell, the zygote. Fertilization. Union of mature male and female gametes to form a single cell zygote, which occurs in the ampulla of the uterine tube, is called fertilization. Cleavage. The series of mitotic divisions of the zygote which results in increased number of cells with granular reduction in size is called cleavage. The resulting cells of the cleavage which become smaller with each division is known as blastomere. Cleavage. So fertilization and then cleavage. Stages of cleavage. The first cleavage division gives rise to 2 blastomeres by mitotic division. These cells divide into 4 blastomeres. Second cleavage, 4 blastomeres and 8 blastomeres. Third cleavage. After the third cleavage, 8 cell, the blastomere maximizes their contact with each other, forming a compact ball of cells held together by tight junctions. This process is termed the compaction. About 3 days, 72 hours after fertilization, cells of the compact embryo divides again at fourth cleavage to form a 12 to 16 cell stage called the morula or the mulberry. 3 days hours fertilization. About 3 days after fertilization, cells of the compact embryo divides again in fourth cleavage to form a 12 to 16 cell stage called morula. 3 days after fertilization, again at fourth cleavage. The inner cell of the morula constitute the inner cell mass of the embryoblast. The surrounding outer cells compose the outer cell of the trophoblast. The surrounding outer cells compose the outer mass of the trophoblast. The inner cell mass gives rise to the tissue of the embryo proper, and the outer cell mass constitute to the placenta. Blastocyte formation. At fourth day, morula enters the uterine cavity. Uterine endometrium is at this time is at secretory phase. Blastomere formation. Third day, at 16 cells called morula. After third day fertilization, cells compact embryo divide into again fourth cleavage. Third day, fourth cleavage and 16 cell storage called morula. Inner surface of the morula constitute of inner cell mass, and inner mass refers to the embryo proper, and outer cell mass constitute to the placenta. Morula. 4 cell stage is morula. At the fourth day, morula enters the uterine cavity. Uterine endometrium at this time secretory phase. Fluid enters the intracellular space and of inner cell mass through the zona pellucida. Gradually, the intracellular spaces become convoluted, and finally, a single cavity, the blastocell or the blastocyte cavity, is formed. At this time, embryo is a blastocyte. The blastocell or the blastocyte cavity is formed. At this time, embryo is a blastocyte. Blastomere in these blastocytes are arranged as follows. Blastocell, single cavity, the blastocell or blastocyte is formed. Blastocell and blastocyte cavity is formed. Embryo is a blastocast. Blastomere in the blastocytes are arranged as follows. Embryoblast. Cells of the inner cell mass at one pole of blastoclast are called the embryoblast. Is given the rise to embryo proper. Trophoblast. Cells of the outer cell mass from the flattened of the four of the epithelial walls of the blastoblast are called trophoblast. It gives rise to the embryonic part of the placenta. Parts of the blastocytes. Embryonic pole. This is the area of attachment between the inner cell mass and the overlying trophoblast. Inner cell mass and the overlying trophoblast. Embryonic pole. It is opposite to the embryonic pole. Zona pellucida disappears at fifth day, allowing implantation to occur. Implantation. Embedding of the blastocytes in the endoderm of the uterus is called implantation. It begins at the first to second week, but converts into second week as the implantation process is completed by the end of the second week. Second week. Define morula. A morula is the embryo at an early stage of embryonic development, consisting of cells, blastomeres, a solid ball containing in the zona pellucida. The morula is produced by embryonic cleavage, the rapid division of zygote. Once the zygote is divided into 12 to 16 cells, it resembles a mulberry, hence the name morula, Latin mulberry. Process of formation of morula. As the zygote passes along the uterine tube towards the uterus, it undergoes a cleavage, a series of mitotic cell division into a number of smaller cells, blastomeres. After the third cleavage, 8 cells, the blastomeres maximize their contact with each other, forming a compact ball of cells held together tightly by tight junctions. This process is termed as compaction. About 3 days after fertilization, cells of the compact embryo divide again for cleavage to form a 12 to 16 cell mass called morula. The morula is composed of two parts, which will become the embryo part and which will contribute to the formation of the placenta, outer cell mass and inner cell mass. Draw and label the structures of blastocyte. Mention the role of inner cell mass and outer cell mass of blastocyte in the development of embryo. Role of inner cell mass and outer cell mass in blastocyte in the development of embryo. Cells of the inner mass at the pole of blastocytes are called embryoblasts and give embryonic proper, and the cells outer cell mass on the flattened are from epithelial wall. So the inner cell mass gives embryo proper, and the outer cell mass of the epithelial wall of blastocytes are called the trophoblast. They give rise to the embryonic part placenta wall, flattened from the epithelial wall of the blastocytes are called trophoblast. It gives rise to the embryo part of the placenta are called trophoblast. Cell mass of the epithelial wall of the blastocytes are called trophoblast. Gives rise to the embryonic part of the placenta. And the cells of outer cell mass flattened from epithelial wall of blastocyte are called trophoblast. It gives rise to the embryonic part of the placenta. Write difference between morula and blastula. Morula and blastula. In the early stage, in the late stage, 3 days after fertilization, 3 to 4 to 5 days from fertilization. Blastocyte. Spherical, rounded and hollow ball like 3 to 4 days after fertilization. Single inner cell mass and outer cell mass. Embryoblast and trophoblast. Blastocyte. Number of cells 12 to 16. Many cells about 70 to 100. Presence, absent. Zona pellucida. Presence of cavity. Absent and presence. Fate. Formation of blastobust implantation. Blastocyte and morula. Draw and level. The events takes place in the first week of the human development. Implantation. Define implantation and the embedding of the blastocytes in the endometrium, now called decidua of uterus, is called implantation. It starts at 5th to 6th day of fertilization and completes at 13th day of fertilization. Implantation. Process of implantation. By the help of the ciliary muscle contraction, the morula developing blastoplast is transported through the fallopian tube. By the developing blastocyte, the transported through the capillary tube. Uterine mucosa already undergoes several changes to receive blastocytes during implantation. The mucosa is the secretory phase of the menstrual cycle. Possesses 3 layers. The polar trophoblast overlaying the embryoblast now adheres to the uterine mucosa and starts penetration into the eroding of the surface. This eroding is due to the proteoblast enzyme secreted by trophoblast. By the help of capillary muscles, morula is transported through the fallopian tube. Uterine mucosa already undergoes several changes in the received blastocytes. During implantation, the morula is the secretory phase and menstrual cycle of 3 layers. Uterine mucosa. Process of implantation. By the help of ciliary and muscular contraction, morula transported. Polar trophoblast now adheres to the uterine mucosa and transports the penetration into the endometrial surface. This eroding due to the proteolytic enzyme secreted of the trophoblast. By eroding of the surface, trophoblast gradually penetrates the endoderm in a spongy layer. This process in the implantation is completed. Normal size of implantation. Posterior wall of the uterus close to the fundus. But implantation anywhere on the upper part of the uterus cavity is considered as normal. But upper part of the uterus cavity is considered normal. Many study tubular, isthmus, tubular, ovarian, internal as an abdominal cavity, normal side. Nice to know. 3 distinct layers can be recognized in the endometrium: a superficial compact layer, a intermediate spongy layer, a thin basal layer. A superficial compact layer, a intermediate spongy layer, superficial compact layer, stratum compactum, and intermediate spongy layer, stratum spongium. 3 distinct layers recognized on the endometrium: a compact layer, a spongy layer, and a basal layer. Mention the abnormal sites of implantation. Ectopregnancy. Implantation of blastocytes in abnormal locations outside the uterus is called extrauterine pregnancy or ectopregnancy. This may occur at the place in the abnormal cavity, ovary and uterine tube. Abnormal locations found outside uterus is called extrauterine pregnancy. Common sites of ectopic pregnancy, ectopic implantation outside the uterus, uterine tube, tubular pregnancy. 95% ectopic pregnancies occur here. Abnormal cavities. These in the rectus cavity of the pouch of Douglas. Abdominal cavity. The mesentery and ovary, primary and ovarian pregnancy within the uterus. Internal opening of the uterine resulting in placenta previa. Fate of ectopic pregnancy. In the most cases, the embryo dies within the second month of ingestion. Case: severe hemorrhage and abdominal pain in the mother. Thighs about second month causing severe pain. Implantation at the internal os results in placenta previa with the attending risk of antepartum hemorrhage. What is most relevant term to describe the condition? Most relevant term describe the condition ectopic pregnancy. The positive pregnancy test indicates pregnancy, but the absence of fetal sac or the pole in the uterine cavity of the ultrasonography is the key finding of struggling ectopic pregnancy, suggesting an ectopic pregnancy. Common sites of such condition and clinical sequence of this condition. Ectopic pregnancy. Implantation, normal implantation sites of ectopic pregnancy. What are the changes occur in the uterine endometrium during implantation? Changes in the uterine endometrium during implantation. At the time of implantation, the mucus of the uterus is secretory phase and responds to the progesterone produced by the corpus luteum. At the implantation, the mucus of the uterus is a secretory phase in the response to the progesterone produced by the corpus luteum. Implantation, during which the time uterine glands becomes coiled. This tissue becomes secluded. As a result of three distinct layers. This can be recognized from the endometrium: a superficial compact layer, intermediate spongy layer, and a basal layer. There are the changes occur in the uterine after implantation. Changes that occur after implantation: increased thickness, increased vascularity, elongated branches in uterine glands. The connective tissue cells of the decidua enlarge from the pale staining decidual cells. They are large rolled cells with one or more nuclei, increased leukocytes, and occurrence of decidua reaction. What is decidua? Draw and label the different parts. Short note: decidua is the functional layer of the endometrium which is shed during parturition. Decidua is the functional layer of the endometrium during parturition. Parts of the decidua: decidua basal, decidua capsulis, and decidua partialis. Decidua basal. The decidua basal from the cranion fronsdom of the decidua basalis consists of a compact dense layer cells, decidual cells, which abundant amount of lipids and glycogens. It forms the maternal portion of the placenta. Decidua capsularis, layer of the embryonic pole, is decidua capsulis. It separates the embryo from the uterine cavity. And decidua partialis, the rest of the decidua. What do you mean by decidua reaction? The cellular and the vascular changes in decidua resulting from pregnancy under the influence of progesterone is referred as a decidua reaction. Changes which are known as the decidua reaction: abnormality glands becomes hypersensitive, endometrial glands become hypersensitive, stromal cells of the endometrium becomes polyhedral and loaded with glycogen and lipids. Intercellular spaces are filled with extravasate and the tissue becomes soft and edematous. These changes at first are confined in the area of intermediate surrounding to the implantation site, but soon occur through the endometrium. Gateway of biochemistry. Solution. Definition: solution is the homogeneous mixture of two or more substances, solid, liquid, or gas, distributed uniformly among each other. Homogeneous mixture. It is a mixture of two or more substances showing all through the same composition. Heterogeneous mixture. It is the mixture of two or more substances showing different composition of different parts of the total mixture. Solvent. Solvent is the component of a solution which forms a large portion of that solution. Physical state of the solvent must be same as the physical state of the total solution. Solute. Solute is the component of a solution which form a minor part of the solution. Solutes are held in a solution by the solvent. For example, 0.9% solution here, water is solvent because it appears in the major portion, and in physical state is liquid like the physical state of a total solution. Here sodium chloride is the solute because NaCl presents minor portions of the solution and it is kept in a soluted by water. Heterogeneous solution. It is a heterogeneous solution is a mixture of solvent and solutes where solutes exist as aggregates of molecules. For example, solutes of protein in water. Homogeneous solutes are true solutes or molecular solution. It is a homogeneous mixture of solute and solvent because where solutes exist as a molecular ion. Here solutes are crystalloid with size less than 1 nanometer and solutes do not sediment out of the solution because even on indefinite standing these solutes cannot be separated from the solvent by passing through the solution through filter paper or semi-permeable membrane. In fact, a solution means mixture of two or more substances. It could be homogeneous or heterogeneous, but traditionally by the term solution we take it to mean the homogeneous solution. It is otherwise known as a true solution. Concentration of a solution. It is the amount of solutes present in a given quantity of solvent or solution. Atom. Atom is the smallest particle of a substance that takes part in chemical reaction. Atom consists of a nuclear composition and protons, positive charged particles and neurons, particles without any charge surrounded by electrons, negative charged particles. Element of a substance made out of only one kind of atom, for example, sodium. Compound is a substance made out of two or more different kinds of atoms, for example, water. Atomic weight. The atomic weight is the sum of number of proton and neutron of an atom. It indicates the number that expresses how many atoms an atom is heavier than one hydrogen atom. For example, atom weight of sodium is 23. What it means is that one atom sodium is 23 times heavier than the one atom of hydrogen. Atomic weight of hydrogen is 0.1. Atomic weight of oxygen is 16. 0.1 and 16. Molecule is the smallest particle of a element or compound that can be normally exist freely. Molecular weight is the sum of atomic weight of all atoms of a molecule. It indicates the number that expresses the how many times a molecule is heavier than one atom hydrogen. Molecular weight of H2O 18, 2 into 1 plus 16. That means one molecular weight is 18 times heavier than the one hydrogen, 1 gram atom HCl. Equivalent weight. The equivalent weight of a substance, element or compound is the number which indicates how many parts by weight of a substance will combine with or displace 1 by weight of a hydrogen and 8 parts of oxygen, or 35.5 parts of the weight of chloride. Oxygen 35.5 parts of chloride. Equivalent weight of calcium is 20. That means 20 parts by weight of calcium will be combined with the or displace 1 part by weight by hydrogen or by 35.5 parts of weight of chloride. Cation, positive charged ion, anion, negatively charged ion, Zwitter ion, dipolar or hybrid ion. Zwitter ions are ionized neutral spaces having equal amount of positively charged and negatively charged groups with net charge 0. Electrolytes. Electrolytes are molecules that dissociate in the water into charged particles called ions, a cation and anion, and capable of carrying electric charges. Important electrolytes are sodium, potassium, chlorine, and carbide, HCO3 minus. Clinically, these four electrolytes are frequently and routinely measured in body fluid in the name of electrolyte profile for day-to-day patient management. Calcium calculates body and the fluid in the name of electrolyte profile for day-to-day patient management are frequently routinely measured in the body. Clinical phosphate and magnesium in ionic formation are also regarded as electrolyte but not routinely measured. Glucose and urea are not electrolyte because they don't dissociate in water and they have no charge. Now an osmole. An osmole, symbolized as OSM, osmole. It is the amount of osmotically active substance in a gram. It is the amount of osmotically active substance in a gram, which is 1 liter of solution with water exerts osmotic pressure of 22.4 osmotic atmosphere and depress the freezing point of water by 1.86 Celsius and depresses the freezing point of water by 1.86 degrees. Okay? It is the amount of osmotically active substance in a gram. It is the amount of osmotically active substance in a gram, which is 1 liter solution with water exerts osmotic pressure. Which 1 liter of solution, which is 1 liter solution with water exerts osmotic pressure of 22.4 atmospheric pressure, that freezing point. 22.4 atmospheric pressure. 1 atmosphere equal to 760 millimeter per mercury. Osmolarity active substance means it is the amount of osmotically active substance in gram. It is the amount of osmotically active substance present in gram present in 1 liter of solution. Osmotically active substance means any substance which can exert osmotic pressure in solution with water. Osmotically active substance means any substance which can exert osmotic pressure in solution with water. Relatively small amount of high osmotically active substance, for example sodium, glucose, etc. Less active substance, for example protein. For less active substance, relatively a big amount of equal into 1 osmole. Osmole is used as the unit of measurement for osmotically active substances. Unit of measurement for osmotically active substances. Units of measurement. Way to express concentration of solution. Percentage by weight. It is gram of solute per 100 gram solution. For example, 5% dextrose means 5 gram dextrose present in 100 gram solution. The percentage by volume. It is the gram of solute per 100 gram solution. It is the gram of solute per 100 gram solution where solution is solid, or it is the ml of solution per 100 ml solution when solute is liquid. 5% dextrose means 5 gram dextrose present in 100 gram solution. 5% alcohol means 5 milliliter alcohol present in the 1 ml solution. Molarity. It is the number of mole of solute per liter solution. It is molarity. It is the number of mole of solute present. It is the number of mole of solute per liter present in 1 molar glucose solution means 1 mole glucose present in 1 liter of solution. 1 mole. Molarity. And molality, the number of mole of solute per kg solvent. Molality is the per kg solvent. 1 molar glucose solute means 5 mole glucose present in 1 kg of solvent. 1 kg of solvent. Normality. It is the number of equivalent of EQ or solute in per liter solution. For example, 2 NaHCl. 2 normal HCl means 2 equivalent HCl percentage of 1 liter of solution. 2 equivalent HCl present of normal 2 equivalent presence means it number of equivalent of solute. Equivalent of osmolarity. It is the number of osmole of solute per liter solution. Number of osmole per Osmol of solute. It is the number of osmol solute related to osmolality. Osmolality. It is the number of osmol or solute per kg of solvent. 3 osmolar glucose solutes means 3 osmotic glucose present in 1 kg of solvent. Chapter 6. Chemistry of enzyme. Catalyst. Substrate that alters the rate of chemical reaction but itself remain unchanged chemically at the end of reaction. This is called catalyst. Positive catalyst enhances the rate of chemical reaction but negative catalyst retards the rate of chemical reaction. Definition of enzyme. Enzymes are biocatalysts produced by living cells to catalyze biochemical reactions. Enzymes are always positive catalysts. So, enzymes accelerate the rate of chemical reaction but itself remain chemically unchanged at the end of reaction. Properties of enzymes. Protein in nature, colloidal and water soluble, non-dialyzable, heat labile. Heat causes destruction of the enzyme and loss of function. Act as catalyst and have definite isosteric pH. Difference between enzyme and hormone. Enzyme and hormone. Absolute protein in nature act at the site of production, do not go to the site of act via blood. Shows catalytic activity. Hormone may be other than protein, for example steroid, usually act distance to the site of production, go to the site of reaction via blood. Shows regulatory activity, controls enzyme activity. Characteristics of enzyme activity. Do not initiate the reaction, accelerate the rate of reaction. After initiating the reaction, after the equilibrium quickly. Do not alter the extent of reaction, equilibrium context of reaction. Final destination of reaction is not shortened, but it is reached quickly. Highly reaction specific. Interact with one or few specific substance and catalyze only one type of reaction. Substrate is the substance on which enzymes act to convert them to product. Remain uncharged chemically in the overall process. Enter into reaction but come out of the reaction without any change. Effective in extremely small amount and functionally independent of parent cell. Activity can be regulated by activation or inhibition as per cellular need. Enzyme specificity. Definition of enzyme specificity. Enzyme activity is sufficient for a specific substrate and a specific type of reaction. Remain unchanged chemically in the overall process. Enter into the reaction but come out of the reaction without any change. Effective in extremely small quantity and functionally independent parent cells. Enzyme specificity. Definition of enzyme specificity. Enzyme activity is specific for a specific substance and a specific type of reaction. Type of enzyme specificity. Substrate specificity. Act on only one substrate or related substrates. Absolute specificity. Act on only one specific substrate. Glucokinase. Act on glucose. Urease. Act on urea. Catalase. Act on H2O2. Relative specificity. Act on structurally related substrate. Trypsin. Act on different proteins. Hexokinase. Act on all hexons. Reaction specificity. One substrate may undergo different type of reaction, but one enzyme catalyze only one type of reaction. One substrate may undergo one reaction. Pyruvate can undergo several reactions to produce different products, but each reaction is catalyzed by a separate enzyme only for the reaction. Importance of enzyme specificity. It marks possible for lots of enzymes to coexist in a cell without interfering each other's action. These are non-protein helper molecules essential for catalytic activity of enzyme. For example, coenzyme and cofactor coenzyme. These are mostly vitamin B complex derivatives. Properties characteristics of coenzyme: non-protein in nature, relatively heat stable, low molecular weight organic substance, mostly vitamin B complex derivatives. Dialyzable, can be separated from the enzyme by dialysis. Usually, coenzymes remain tightly bound to enzymes, so coenzymes are often called prosthetic group or co-substrate of enzyme. Enzyme that bind with coenzyme is called haploenzyme, and the total complex composed of apoenzyme or coenzyme is known as holoenzyme. So, holoenzyme is a conjugated protein which is the active form of coenzyme, apoenzyme, because without coenzyme, apoenzyme is alone is inactive. Coenzyme derived from vitamin B complex, for example, NAD/NADP, niacin, FAD/FMN, riboflavin, CoASH, pantothenic acid, pyridoxal phosphate, pyridoxine B6, thiamine phosphate, TTTP, thiamine B1. Non-vitamin coenzymes, for example, coenzyme Q, lipoic acid, bipoteirin S, aldosterone, methionine SAM. Cofactors, these are usually trace elements. Cofactors are usually inorganic metallic ions which help in enzyme activity. Most of these cofactors remain loosely bound to enzyme when the enzymes are called metal activated enzymes. These less frequently contractile found to remain tightly bound to the enzyme when the enzyme are called metalloenzyme, and cofactors are called prosthetic group, for example carboxypeptidase and zinc. Enzymes and cofactors. Enzyme: carbonic anhydrase, alkaline phosphate, and DNA polymerase, RNA polymerase. Cofactor: zinc, hexokinase. Cofactor: magnesium, catalase and tyrosine. Cofactor: copper, catalase and peroxidase. Cofactor: iron, xanthine oxidase, molybdenum. Lipase: calcium, amylase chloride. Active site: it is the small area of enzyme which binds substrate for catalysis. It is the small area of coenzyme which binds substrate for catalysis. Active site creates three-dimensional cleft like conformation complementary to the substrate conformation for easy substrate binding. Allosteric site: these are specific areas on the surface of enzyme other than the active site. Different allosteric effectors. Allosteric modifiers bind with the enzyme through allosteric site to modify and regulate enzyme activity. Allosteric sites. Enzyme units of enzyme activity. Expression of enzyme activity: 1 IU. International unit of enzyme is the amount of enzyme required to convert 1 micromolecule substance to 1 micromolecule product per mixture under optimum condition, and it is expressed as 1 IU. 1 IU equal to 1 micromole per minute. 1 kettle of enzyme cat is the amount of enzyme required to convert 1 molecule substance to 1 molecule product per second under optimum condition. So 1 cat equal to 1 molecule per second. IUBMP classification of enzyme. It is an uniform and unambiguous enzyme nomenclature system adopted by International Union of Biochemistry and Molecular Biology. It has been done according to the type or mechanism of chemical reaction catalyzed by the enzyme. This classification has been in use since 1964. IUBMB classified enzymes into 6 main classes. Under every class, number of different subclasses, and under every subclass, number of different subclasses proposed. Finally, under every subclass, all relevant enzymes are numbered serially. Class indicates the type of reaction, subclass indicates the type of group involved in the reaction, and subclass denotes the substrate on which the enzyme acts. Following 6 main classes of enzymes are proposed on IUBMB classification. Oxidoreductase. This group of enzymes catalyze the oxidation and reduction of the substrate. For example, AH2 plus B equal to B plus BH2, dehydrogenase and oxidase. Transferase. This group of enzyme catalyzes the transfer of a particular functional group from one substrate to another. For example, alanine amino transfer. Hydrolase. This group of enzyme catalyzes the hydrolysis of substrate. They do not need any coenzyme. For example, lactase and sucrase. Lysis. This group of enzyme catalyzes the splitting of the large substrate with removal of a small group from the substrate by non-hydrolytic cleavage, leaving a double bond on the substrate in most of the cases without ATP expenditure. For example, decarboxylase, aldose. Isomers. This group of enzymes catalyzes the isomerization and formation of isomer of a particular substrate. For example, isomerase and epimerase. Ligases. This group of enzymes catalyzes the linking of two molecules by covalent bond without expenditure of ATP. For example, DNA ligase and carboxylase. Factors affecting coenzyme activity: temperature, pH, substrate concentration, enzyme concentration, product concentration. Now, Meckel's constant Mn of an enzyme is the substrate concentration at which enzyme shows half maximum velocity. Constant Km of an enzyme is the substrate concentration at which enzyme shows half maximum velocity. At this point, 50% of enzyme binds with the substrate. Km indicates the enzyme substrate's affinity, and it is inversely proportional to the enzyme affinity for substrate. The more the enzyme affinity for substrate, Km value will be low. That means substrate at low concentration will show half maximum velocity. The less the enzyme affinity for substrate, Km value will be high, and substrate must be high concentration to show the half maximum velocity. So Km provides a measure of the substrate concentration required for significant catalysis to occur. Km value is concentration for a given enzyme at a defined condition. Velocity of enzyme activity maintains hyperbolic curve with substrate concentration. Inhibition of enzyme activity. Types of inhibition. Reversible inhibition. Here inhibition shows structural symmetry with substrate and components with the substrate for the same activity of the enzyme. Non-competitive inhibition. Here inhibitor is structurally unrelated to the substrate and inhibitor binds with the enzyme at the allosteric site. Irreversible inhibition. Here inhibition of the structure unrelated to the substrate and inhibition binds with the enzyme at the allosteric site and inactive enzyme. Application of enzyme inhibition. Treatment of a disease, study of metabolic pathways, study of mechanism of enzyme action. Competitive inhibition and non-competitive inhibition. Binds with active site of enzyme, binds with allosteric site of enzyme. A structurally substrate analog, a structurally unrelated to substrate. Cause irreversible inhibition, mostly cause irreversible inhibition. Inhibition related by increasing substrate concentration. Inhibition not related by relieved by increasing substrate concentration. Reduce the affinity of enzyme to substrate. Enzyme substrate affinity is not altered. Most concentration with drug action, mostly concerned with toxicological use. Regulation of enzyme activity in biological system in vivo. Compartmentation of substrate invaluability. Allosteric regulation act within minutes. Covalent modification of enzyme act within the minutes to hours. Conduction and repression of enzyme act within hours to days delayed. Feedback inhibition. Activation of inactive enzyme, zygoenzyme. Clinical importance of enzyme. Enzyme deficiency cause disease, for example, lactose intolerance, lactase, hemolytic anemia, glucose 6-phosphate, glycogen storage disease, enzymes of glucose metabolism, lysosomal storage disease, lysosomal enzyme. Enzymes are used as diagnostic tool for disease, liver disease, obstructive liver disease, isomeric heart disease, muscle disorder, prostatic cancer, bone disorder, acute pancreatitis. Enzymes are used as prognostic tool of disease. Enzymes are used as therapeutic tools of disease. Enzymes inhibition is used to therapeutic tool of disease. Use of enzyme as laboratory tool of diagnosis of genetic disease. Isomers. Creatine phosphokinase, lactate dehydrogenase, alkaline phosphatase, HMG coenzyme, carbamyl phosphatase, nitric oxide synthesis, cytochrome P450 monooxygenase, hexokinase, cyclooxygenase, amino levulinic acid synthesis 1, glutaminase. Lung alveolus is lined by simple squamous epithelium. Simple squamous epithelium is needed in places where there is no need for absorption, secretion, or ionic transport, and where the need for any exchange by the process of diffusion. Lung is an organ of gaseous exchange, and this exchange occurs in alveoli of lung by the process of simple diffusion through respiratory membrane. One of the layers of respiratory membrane is simple squamous epithelium, which lines with alveoli. According to law of diffusion, the thinner the diffusion membrane is, the better will be the rate of diffusion. Simple squamous epithelium is the thinnest of all the epithelium. So, for effective gaseous exchange by diffusion, lung alveolus is lined by simple squamous epithelium. Explain anatomically, histologically. Capillaries are lined with simple squamous epithelium. Simple squamous epithelium in lined blood vessels. Simple squamous epithelium covers the surface that allows passive transport of gases and fluids. In the blood vessels, this epithelium or endothelium allows passive transport of fluid, nutrient, and metabolics across the thin capillary walls. For this simple squamous epithelium line blood vessels. Explain anatomically, histologically why stomach is lined by simple squamous epithelium. Simple columnar epithelium. Simple columnar epithelium covers the surface of the stomach. These cells are secretory and produce mucus. The mucus covers the stomach surface and protects its surface lining from the corrosive gastric secretions that are normally found in the stomach during food processing and digestion. Simple columnar epithelium lines most part of the gastrointestinal tract. Simple columnar epithelium lines most part of the gastrointestinal tract. These cells are secretory and produce mucus. The mucus covers the stomach surface and protects its surface lining from the corrosive gastric secretions that are normally found in the stomach during food process and digestion. Some of the columnar cells lining the GI tract are specialized by processing brush borders, which are formed by numerous microvilli. They help in active absorption of sugar and other chemical substances. So, simple columnar epithelium lungs most part of the gastrointestinal tract serves the function of protection, lubrication, absorption, and secretion. Explain anatomically, histologically why fallopian tubes is lined by simple ciliated columnar cells. Fallopian tube is lined by simple ciliated columnar cells. The fallopian tube or alveolar duct is lined by simple organelle cells which facilitate the conduction of oocyte and sperm across the surface. The epithelium contains two functional important cell groups, ciliated cells and non-ciliated secretory cells or Bragg cells. The cilia beats towards the uterus. The heavily ciliated cells on the febrile surface create a current that guides the oocyte into the infundibulum of the uterine tube. The cilia actions of the muscular contractions in the wall of the uterine tube transports that captured oocyte or fertilized egg through the remaining region of the uterine tube towards the uterus. Explain anatomically histologically why the small intestine epithelium contains villi and goblet cells. The small intestinal epithelium occurs villi and goblet cells. The main function of the epithelium is the small intestinal absorption. This function is enhanced by the process of finger-like villi, which increases the absorption surface area and which are covered by simple columnar epithelium with striated borders or microvilli. This absorbs nutrients and fluids from the intestinal contents. The intestinal epithelium also contains numerous goblet cells. These cells secrete mucus, which protects the surface lining from the corrosive secretion that enters the small intestine from the stomach during digestion. Explain anatomically, histologically, developmentally goblet cells are numerous in large intestine. Goblet cells are numerous in large intestine. Digestion and absorption of essential end products of the digestion occurs completely in small intestine. So the intestine contents that enter the large intestine contains mainly water and electrolytes. In addition to waste material, the function of large intestine is to absorption of water and essential electrolytes as far as possible and conversion of liquid primary contents into semi-solid formed fecal matter. Due to the absorption of water, the contents become gradually hard and solid. There is a chance of friction between the solid and hard large intestinal contents with its lining epithelium, so it gives protection to the large between the solid and the hard large intestine contents with its lining epithelium, so gives protection of the large intestinal wall. Large intestine contains progressively increasing amount of goblet cells that secrete mucus. Digestion and absorption of essential and protodigestion occurs completely in a small intestine, so it gives protection and large intestinal wall. Large intestine contains progressively increased amount of goblet cells that secrete mucus. Explain anatomically, histologically why proximal coagulated tubes of the kidney are covered with brush border or microvilli. Proximal coagulated tubules of the kidney are covered with the brush border or microvilli. Production of urine by the kidney involves filtration, absorption, and secretion. The apical surface of the simple cuboidal epithelium in the proximal coagulated tubules of the kidneys are also covered with brush border or microvilli. The main function of this microvilli is to absorb the nutrient material and fluid from the filtrate that passes through the tubules. Explain from your knowledge of anatomy why esophagus is limited lined by non-keratinized stratified squamous epithelium. Esophagus is lined by non-keratinized stratified squamous epithelium. Esophagus is an upper part of the alimentary tract responsible for the passage of partly or completely chewed food through the pharynx or the stomach. Esophagus is only a food passage having no other function. Mucous membrane of esophagus is subjected to the friction by food, which can be soft or hard, hot or cold. For protection of its wall from injury by food, esophagus is lined by a stratified epithelium. As no digestion and absorption occurs in esophagus during passing of the food, there is no need for secretion of any digestive juice containing any enzymes protecting in nature. The lining epithelium cells have less amount of cytoplasm containing fewer organelles, so these cells remain squamous type. As esophagus lies within interior of the body, its mucus is not exposed to air. Thus, its lining epithelium is not non-keratinized. So, esophagus is lined by non-keratinized, stratified squamous epithelium. Explain anatomically. Histologically, oral cavity is lined by non-keratinized epithelium. Oral cavity is lined by non-keratinized squamous epithelium. In the oral cavity, food is digested, masticated, chewed, and lubricated by saliva for swallowing. Mucous membrane of oral cavity is subject to friction by food, which can be soft or hard, hot or cold. For protection of its wall from injury by food, oral cavity is lined by a stratified epithelium. As no digestion and absorption occurs in oral cavity during passing of the food, there is no need for secretion of any digestive juice containing any enzymes, that is, protein in nature. The lining epithelium also has less amount of cytoplasm containing fewer organelles, so the cells remain squamous in shape. As esophagus lies within the interior of the wall, its mucosa is not exposed to air. Thus, its lining epithelium is non-keratinized. So, oral cavity is lined by non-keratinized, stratified squamous epithelium. Explain anatomically, histologically how transitional epithelium prevents absorption. Transitional epithelium prevents absorption of the urine. Transitional epithelium, also called urothelium, is a specialized form of stratified epithelium lines the major portions of urinary tract, including the urinary bladder. It prevents reabsorption of urine because of its some special characteristics. It's multi-layered. Superficial cells are dome-shaped, called umbrella cells, having the capacity to be stretched or relaxed. Luminar surface of superficial cells are covered by a glycolipid coat consisting of cerebrocyte, which acts as urine-proof barrier. Glycocalyx propels away urine. Presence of light junctions between adjacent cells of specific layers. Explain from your knowledge of anatomy why transitional epithelium layers urinary bladder. Urinary bladder is lined by transitional epithelium. Transitional epithelium, also called urothelium, is a specialized form of a stratified epithelium lines the major portions of the urinary tract, including the urinary bladder. Functions of transitional epithelium in the urinary bladder: prevents reabsorption of urine. Superficial cell layers of the transitional epithelium of urinary bladder consist of large, sometimes multi-nucleated umbrella cells which are connected by each other by tight junction and form a permeable layer to prevent reabsorption of urine. Cause the urothelium into the bloodstream. Allows free distension and relaxation of bladder mucosa. Polygonal cells of the intermediate cells of transitional epithelium process the capillary to stretch and flatten, which allow the distension of transitional epithelium as the urinary bladder fills. Explain anatomically, histologically. Development. Skin contains keratinized stratified epithelium. Skin contains keratinized specialized squamous epithelium at places where the epithelial surface is dry, as in the skin surface due to the dehydration of keratinized, rich in teratinocytes or contrast. The skin lacks nuclei. These forms of non-keratinized covers the epithelium. This kind of epithelium, described in keratinized stratified epithelium, is the thin, metabolically inactive surface of the cells of the epithelium helps protect against water loss across the epithelium. Living epithelium of various organs and glands. Lips and gums. Keratinized stratified squamous epithelium. Squamous. Oral cavity. Cheeks. Non-keratinized stratified. Squamous. Hard plate, soft plate. All of them are squamous. Tongue. Non-keratinized stratified squamous epithelium. Pharynx. Non-keratinized stratified squamous epithelium. Esophagus. Non-keratinized stratified squamous epithelium. Stomach and pectinate line of the canal. Simple columnar epithelium. Stomach and pectinate line of the anal canal. Columnar epithelium. Esophagus. Non-keratinized stratified squamous epithelium. Pectinate line of Hilton's white line of anal canal. Non-keratinized stratified squamous epithelium. The squamous, columnar squamous, stratified squamous epithelium. Pseudo-stratified ciliated columnar epithelium. Except nasopharynx, which is columnar epithelium. Lower part of anal canal below Hilton's white line. True skin of keratinized stratified squamous epithelium. Nasal cavity, vestibule, keratinized squamous. Respiratory columnar, pseudostratified. Olfactory epithelium, columnar squamous. Pseudostratified columnar squamous, columnar squamous, columnar cuboidal squamous. Urogen system, the urogenital system. Bowman's capsule, simple squamous. Proximal covulated tube, cuboidal epithelium and brush border. Thin segment, simple squamous epithelium, simple cuboidal epithelium. Distal covulated tube, simple cuboidal epithelium with few microvilli. Cuboidal. Basement membrane. Basement membrane. The term basement membrane used to specify a periodic and acid sniff (PAS) positive layer visible with the light microscope beneath epithelial structure. The basement membrane is formed by the combination of basal lamina and reticular lamina and is therefore thicker. Basal membrane. A felt-like sheet of extracellular material at their basal surface of all epithelial cells in contact with subjacent connective tissue is called basal lamina. The structure is visible only with the electron microscope, where it appears as an electron-dense layer, 20 to 100 nanometers thick. The dense layer or lamina densa. The clear layer or lamina lucida rara between epithelia with no intervening connective tissue, such as in lung alveoli, in renal glomeruli. In basal lamina is often thicker due to the fusion of the basal lamina for each epithelial layer. Composition. Collagenous IV glycoproteins, laminase, antacid, nidogen, proteoglycans, heparyl sulfate, chondroitic sulfate, and dermatan sulfate. Reticular lamina. Basal lamina are attached to reticular fibers and made of type 3 collagen in underlying connective tissue by anchoring fibers of type 2 collagen, type 3 collagen, and type 7 collagen proteins produced by the cell of the connective tissue and form a layer below the basal lamina called the reticular lamina, and it is also visible by TEM or electron microscope. Functions of basal membrane, basal lamina. Structures and filter functions influence cell polarity, regulate cell proliferation and differentiation, influence cell metabolism and survival, organize the proteins in the adjacent plasma membrane, serve as pathways for cell migration. Why epithelium does not detach the basement membrane? Epithelium does not detach the DM due to presence of hemidosome. Why basement membrane does not detach from underlying connective tissue? Basement membrane does not detach from underlying connective tissue due to presence of anchoring fibers which connect BM to underlying connective tissue, intracellular adhesion, and other junctions. Intercellular adhesion and other junctions. Mentions of the type of intercellular junctions. Intercellular junction. The junction between two adjacent cells is called intracellular junction. Functions of intracellular junctions. Various functions serve to functions as seals to prevent the flow of material between the cells. Sites of adhesion or encroaching junctions. Channels for communication between adjacent cells. Occluding junction cells preventing flow. Sites of adhesion, adhesive or encroaching junctions, and gap junctions. Classification of intracellular junctions. Occluding junction, tight junction or zonula occludens, adhering junction or zonuli adherence, macula adherence, and fascia adherence. Communicating junctions, gap junctions or nexus. Zonula occludens, tight junction or zonula occludens. Singular zonula occludens are the most apical of junctions. Here the adjacent membranes appear tightly opposed or fused. Functions. The apical function of the tight junction is to form a seal that prevents the flow of material between apical cells. The paracellular pathway in other direction. It also helps in prevent the integral membrane of proteins of the apical surface from being transferred to the basolateral surface and vice versa. This allows the two sides of the epithelium to maintain different receptor and function differently. Zonula adherence. This junction also encircles the cell, usually intermediately below the zonula occludens, and provides for the firm adhesion of one cell to neighbors. Desmosome or macula adherence. The desmosome is a disc-shaped desmosome. Zonula adherence and function. Zonula occludens. Tight junction. From seal. Maintain different receptors and function differently. Membrane proteins. A zonula adherence. Immediately below the zonula occludens, it prevents the adhesion. It provides adhesion for cell and its neighbors adhesion. And desmosome or maculata adherence. Desmosome is a disc-shaped structure for the surface of the cell. It is matched with an identical structure at the same surface of the adjacent cell. Disc-shaped adhesions. Function. Desmosomes provide a firm adhesion among the cells. Fascicle adherence. Three adhesive structures bind with the cell to the basal lamina. Facial adherence. These adhesive structures bind with the cell to the basal lamina. Basal lamina function. Desmosome provide a firm adhesion among other cells. Gap junction. Gap or communicating junctions can occur almost anywhere along the lateral membrane of epithelial cells. They appear as regions where adjacent cells' membrane are closely opposed. The protein of gap junctions called connexins from hexameric complex called connexons, each of which has a central hydrophilic pore about 1.5 nanometers in diameter. Function. Gap junction permits the rapid exchange between cells of molecular with small 1.5 nanometer diameters. Some molecules mediating single transductions such as cyclic AMP, cyclic GMP ions move readily through gap junctions, allowing cells in many tissues to act in a coordinated manner rather than in an independent unit. A good example is heart muscle, where abundant gap junctions are greatly responsible for the heart's coordinating beat. Functional syncytium. Sites where gap junctions are found: cyclic and smooth muscle, liver and kidney, skin, urinary bladder, thyroid and adrenal glands, pancreas. Nice to know. Terminal bar. ZO, ZA and MA are tied together terminal bar. Junctional complex. ZO, ZA, MA and JJ are together called JC. Terminal web. Network of actin filament present in apical region of epithelial cell is called terminal web. Explain why there is a lack of cohesion commonly seen in malignant cells. Lack of cohesion. There is a lack of cohesion in malignant cells. Malignant cells remain attached to each other by several adhesive molecules like coadhesives, and also attached to the basement of the membrane by integrins, another adhesive molecules in several sarcinomes. There is down regulation, decrease of cadherins, integrins, and presumably reducing the cohesive of the malignant cells. The cells become loose and this facilitates their detachment. Surface modification of the cells. Surface modification of cells. Typical surface modification. Motile modification. Cilia. Flagella. Non-motile modification. Microvilli and sterovilli. Lateral surface modification. Plications, intracellular junctions. Basal surface modification. Basal enfoldings. Hemidesmosome, basal lamina. Macrovilli. Macrovilli are non-motile finger-like projections on the apical surface of the epithelium. They consist of closely packed bundles of actin microfilament linked by fascin, aspin, and fibrin. They form saturated border in the intestinal absorption cells and brush border in the kidney tubular cells. Macrovilli. Stereocilia. Stereocilia are usually long, branched, immotile macrovilli. Like macrovilli, stereocilia are supported by internal bundles of actin microfilaments that are cross-linked by fibrin. Stereocilia. Actin microfilaments that are cross-linked by fibrin. Cilia are motile hair-like projections of apical cell membrane consisting of a core cytoplasm supported by bundle of microtubules. Distribution functions. Cilia, respiratory uterine tube. Flagella, cell tail of a sperm. Microvilli, a small interstitial epithelium. Gallbladder epithelium, cells of proximal lunar tube. Stereocilia, vas deferens epithelium and ducts epididymis, epithelium, stereocilia. Give the structure, location, and functions of microvilli. Microvilli, finger-like cytoplasmic projections, apical surface. Structure, closely packed bundles of actin martin filaments linked by fascin, epicene, and fimbrine. Sites, a small intestinal epithelium, gallbladder, and epithelium. Cells of proximal renal tube. Functions, they greatly increase the area of cell surface. Microvilli, small intestinal, contain enzymes connected to digestion and absorption. Explain anatomically how cilia is present in the respective tract. Cilia are present in the conducting zone of the tracheobronchial tree. Cilia is present in the conducting zone of tracheobronchial tree because propelling action of the cilia removes the foreign particles towards the exterior. Mention the difference between cilia and stereocilia. Difference between cilia and stereocilia. Cilia, stereocilia. Definition, motility, structure, movement, function, size. Cilia, stereocilia, non-motile. No such movement occurs. Vast difference, for example, ducts of epididymis, secondary air or cells in the internal air. Explain why stereocilia is not cilia. Stereocilia is not true cilia because unlike true cilia, they are made of actin microfilament rather than microtubules. Actin microfilament. They lack the characteristics of 9+2 microtubule arrangement and basal body found in the true motile cilia. Instead, stereocilia are structurally and functionally more similar to macrovilli. They are long, non-motile, and involved in functions like sensory detentions, for example in the inner ear cells, or absorption, i.e. in the epididymis. True cilia, by contrast, often serve in micro movement or sensory signaling and are based in the microvilli skeleton extending from the centrosome. The term secretocilia is a misnomer as they are shared more with macrovilli than the cilia. Secretocilia, so it's a misnomer. Macrovilli than cilia. Give the difference between cilia and microvilli. Core of actin microfilaments, shorter, non-motile, increased surface area for microabsorption, small intestine, epithelium, gallbladder, and epithelium cells proximal to renal tubule. Core of microtubules, relatively longer, motile movement, respiratory, uterine tube, etc. Mention the difference between cilia and flagella. Shorter, multiple, more structured, longer, single moved. Mention the difference between villi and microvilli. Leaf-like projection of the lamina, 0.1 millimeter. Villi is composed of epithelium, lamina, central, lateral. Small intestine, gallbladder, microvilli. Villi are microvilli. Surface modified, it is not a cell surface modification. Causes the 10-fold increase in surface area. Causes a 44 increase in surface area. Cell surface modification, small intestine. Actin microfilaments. Villi is composed of epithelium. And microfilaments linked by they consist of closely packed bundles of actin microfilament. Epithelium. What do you mean by motile cilia syndrome? Motile cilia syndrome. Motile cilia syndrome is a disorder that causes infertility in men and chronic respiratory tract infections in both sexes. Symptoms: immotile spermatozoa, male affertility, and chronic respiratory infections caused by the lack of cleansing action of the cilia in a respiratory tract cause. It caused by immobility of cilia and flagella, induced in some cases by deficiency of dynein, a protein normally present in the cilia. Dynein. Glandular epithelium. Define gland and mention its functional components. Gland. A gland is a large complex aggregates of glandular epithelial cell specialized for secretion or excretion. General structure of a gland. A multicellular gland has two parts. Parenchyma. It is formed by glandular epithelium. In case of exocrine glands, secretory and piece, and conducing protein, duct system. In case of endocrine glands, follicle or cords or clamps, stroma. This is formed by connective tissue, capsule. It surrounds the gland, septa. They divide the gland into lobes and lobules. Intralobular loose connective tissue. Classification of glands according to cellular arrangement. Simple, straight, tubular, grape of Lieberkühn. Simple called tubular, sweat gland. Simple branched tubular, olfactory Bowman's gland. Simple alveolar acnea, cardiac gland. Compound tubular, cardiac gland. Compound alveolar, pancreas. Compound tubular, submandibular, sublingual gland. Compound tubular, submandibular and sublingual gland. According to the nature of secretion. Parotid gland, pancreas, mucus, unicellular goblet cells, multicellular palatine glands, seromucus, sublingual gland, sublingual gland. Parotid gland is multicellular, mucus, seromucus, parotid gland. According to the node of secretion, holocrine, the cells themselves become the secretory products. For example, sebaceous gland of the skin. Aplocrine, discharge apart from the secretory cells as the secretory products. For example, mammary glands. Macrocrine, secretions by exocytosis without any loss of cellular components. For example, pancreas. Cells themselves become secretory. Apocrine, discharge part of the secretory and without the loss of any cellular components. Mammillary gland, skin, sebaceous gland and pancreas. According to the development, ectodermal glands of the skin, endodermal kidney, mesodermal kidney, endodermal thyroid, parathyroid, thymus, liver, pancreas. Define exocrine gland. Exocrine glands are those which secrete their products to ducts into a surface. General structure of endocrine gland. Endocrine glands have ducts. Inside the gland, the ducts run through connective septa and branches repeatedly until the smallest branch and the end of the secretory portion of the gland. Parenchyma of exocrine glands can be divided into the secretory components portion of secretory and pieces, tubular, actinar or alveolar. The conducting portion of the duct system, branched compound glands, unbranched simple gland. The conducting portion, duct system, branched, unbranched. The stroma of exocrine gland consists of capsule, septa, intracerebellar loose connective tissue. Describe in brief the unicellular gland. For example, goblet cell. The goblet cells, those are modified columnar epithelial cells and contain mucinogen granules between the nuclei and the free substances in the cell. Goblet cells are the primary example of unicellular endocrine gland. They produce mucus, oval shape distribution. Goblet cells are positioned among the columnar or pseudo-stratified columnar cells lining the gastrointestinal tract portion of the respiratory tract. Goblet cell structure: their thin basal region sits on the basal lamina, whereas their expanded apical portion, the theca, faces the lumen of the digestive tube or respiratory tract. The theca is filled with membrane-bound secretory granules, whose secretion press the nucleus towards the deep part until the contents of the discharge from the free surface of the cell. The mucus secretion of the goblet cell is manufactured by the rough surface endothelium reticulum, stored and excreted by the smooth surface vesicle of the Golgi apparatus. Function: lubrication and protection of the epithelial sheath of the digestive and respiratory tract. The mucus secretion of goblet cells is manufactured by the rough surface endothelium reticulum, stored and excreted by the smooth surface vessels of Golgi apparatus. Lubrication and protection of the epithelial sheath of digestive respiratory tract. What is myoepithelial cell? Myoepithelial cell, those are the smooth muscle-like epithelial cells possessing contractile properties. Shape: stele or spindle-shaped. Size: several exocrine glands. Sweat, lacrimal, salivary, and mammillary glands. Structure: these cells are located between basal lamina and basal pole of tractary ducts. Alveolar ducts are more longitudinally angled. Myoepithelial cells are connected to each other area and epithelial cells by both gap junction and mesosome. These cells are specialized for contraction, connecting myosin and large number of actin filaments. Function: their major function is to contract around the secretory or conducting portion of the glands, thus help propel secretory products in the duct. They are developed from the ectoderm. What do we mean by serous and mucus acne? These acne produce a watery enzyme-rich secretion. This secretion is typically clear and contains enzymes such as malase, lipase, and protease. Such acne are commonly found in pancreas and parotid salivary glands. Mucus acne. These acne produce thick, viscous secretions known as mucus. Mucus is composed of glycoproteins called mucins, which produce lubrications and protections of epithelial surfaces. Mucus acne are found in glands such as submucosal gland and the digestive tract of mucus glands in respiratory tract. Difference between serous and mucus acne: the smaller, larger, pyramidal in shape, cuboidal or columnar in shape. They form a spherical mass of cell called achinous. Achinous form organized tubular rather than acne. Rounded, present centrally, flatted, present basally. Deep stained nucleus, lightly stained nucleus. Serous cells produce digestive enzyme and other proteins. Mucus cells produce mostly mucosins. Watery, viscous, slimy secretions. Parotid gland, pancreas, sublingual glands. Parotid gland is a serous acne, but sublingual mucus acne. Mucus gland. Write the peculiarities of nerve supply and sweat gland. Usually all postganglion sympathetic nerves are adrenergic, but the exception of sweat glands. Those postganglion symptoms nerve are cholinergic. This is particularly of nerve supply and sweat glands. Enumerate the endocrine glands in the body. Endocrine glands are produced of hormones, which are generally polypeptide or lipid derived from factors that are released into the intestinal fluid. Endocrine glands in the body: endocrine part of hypothalamus, parotid gland, thymus and pineal gland, islets of Langerhans, pancreas, adrenal and suprarrenal gland, testes in a case of male, ovaries in the case of female. They are all endocrines. Also, some temporary glands present in pineal gland, thymus, corpus luteum, and placenta in case of pregnant female. Enzymes. Enzymes. Define enzymes. Enzymes are biocatalyst products produced by living cells to catalyze chemical reactions. Classification. According to IUBMB, enzymes are classified into 6 major groups based on the type and the mechanism of chemical reaction catalyzed by the enzyme. Oxidoreductase. This group of enzyme catalyzes the oxidation and reduction of their substance. For example, oxidase and peroxidase. Transferase. This group of enzyme catalyzes the transfer of particular functional group other than hydrogen for one substrate to another. Hydrolase. This group of enzyme catalyzes the hydrolysis of substrate. They do not need any coenzyme. Lactase, sucrase. The group of enzyme catalyzes the splitting of a large substrate with removal of a small molecule by non-hydraulic cleavage, mostly leaving doubled bond. Isomers. This group of enzymes catalyzes the isomerization and formation of isomers of a particular substrate. Isomers and epimers, etc. Ligase synthesis. This group of enzymes catalyzes the linking of two molecules by covalent bond without expenditure of ATP. Example: DNA ligase, carboxylase, etc. IUB means International Union of Biochemistry and IUBMB means International Union of Biochemistry and Molecular Biology. Here, oxidoreductase example is lactate plus NAD, lactate plus NAD, lactate dehydrogenase, pyruvate plus NADH plus, lactate plus NAD equal to pyruvate plus NADH H plus, lactate dehydrogenase. Transferase, it is acetyl CoA plus choline or acetyl choline plus choline, so choline acetyl transferase acetyl chlorine CoA or glucose plus ATP. Hexokinase equal to glucose 6PO4 plus ADP. Hydrolase, for example, protein plus pepsin HCl equal to peptone. Sucrase plus H2O equal to sucrose and fructose. Lysis includes pyruvate, decarboxylase FAD equal to ethanol as CO2. Glucose 6-PA phosphatase anisomer is equal to fructose 6-P. And pyruvate carboxylase is oxaloacetate. Write down the important properties of enzyme. Properties of enzyme. Enzyme is protein in nature, colloidal, non-dialyzable, heat labile, and water soluble. Like protein, it has definite isoelectric pH. It is biological catalyst. It is not destroyed or permanently altered after reaction. Enzyme has catalytic reaction, active site for reaction. Enzyme has got allosteric site for allosteric molecules. It lowers the energy barrier, activation energy for reaction. Enzymes are highly specific, interacting with one or few specific substrates and catalyzing only one type of chemical reaction. Some enzymes require non-protein auxiliary substance called coenzyme cofactors for the activity. Enzymes can be regulated, that is, enzyme can be activated or inhibited so that the rate of product formation responds to the need of the cells. Many enzymes are located in specific organelles with the cell For example, enzymes for glycolysis are located on cytosol, so glycolysis cannot take place in mitochondria. The glycolysis lysosomes. Write down the characteristics of enzyme activity. Characteristics of enzyme activity cannot initiate the reaction, accelerate the rate of reaction after initiation, cannot alter the extent to which the reaction occurs. Enzyme lowers the energy barrier for a reaction. Lowers the energy barrier for a reaction. Enzymes don't affect the equilibrium constant of a reaction. Highly reaction specific. Remain unchanged chemically at the end of the reaction. Remain unchanged chemically at the end of reaction. Effective in very small amount. Some enzymes require non-protein auxiliary substance for their activity. Mention 4 differences between enzymes and hormones. Absolute protein in nature may be other than protein, for example, steroid. Act at the site of production, usually act on distance to the site of production. Do not go to the site of activation via blood. Go to the site of activation via blood. Shows catalytic activity, shows regulatory activity. How enzyme act. Mechanism of enzyme action. Catalysis of the prime function of enzymes. For any chemical reaction to occur, the reactants must have sufficient activation energy to reach the transition state of active site from where they are converted to products. Activation energy is the energy required by the reactants to undergo the reaction. According to Michaelis-Menten equation, enzyme binds with substance. Formation of enzyme-substance complex. Enzyme lowers the activation energy for the substrate. More substrates reach the transition state due to the low activation energy. More products are produced. Rate of reaction is increased. Do you mean by enzyme efficiency? Enzymes are highly specific, interact with one or a few substrates, and catalyze only one type of chemical reaction. Type of enzyme specificity: stereo specific or optical specificity, reaction specificity, substrate specificity. What is turnover number? The number of substrate molecules converted to produce per enzyme molecule per second is called turnover number. Typically, each enzyme molecule is capable of transforming 100 to 1000 substrate molecules into products each second. Special sites on enzyme molecule. Define allosteric site, allosteric factor, allosteric effects, and allosteric enzymes. Allosteric site: the site of an enzyme which are spatially and physically distant from the catalytic site of the enzyme are called allosteric sites. Allosteric means occupying other space. Allosteric effector: allosteric sites bind with the compounds of other substances. The compounds in the binds with the allosteric sites are called allosteric effectors. Two types: negative effects and positive effectors. Binding of an allosteric effector at the allosteric site causes unconformational change at the catalytic site, leading to alternation in enzyme activity. This is called allosteric effect. Allosteric enzymes. Allosteric enzymes are those whose activity at the active site may be modulated by the presence of effectors at the allosteric site. Write down about active site. Enzyme molecules contain a specific pocket of cleft called the active site, which is concerned with binding of substrate and catalysis. Substrate and catalysis part. Generally, the active site possesses a substrate binding site and a Lytic site. Substrate binding site, catalytic site. Enzyme molecules contain a special pocket or cleft called the active site, which is concerned with binding of substrate and catalysis. Mechanism of action. The active site contains amino acids chains, amino acid side chains that create a three-dimensional surface. Three-dimensional surface complementary to the substrate. The active site binds the substrate, forming an enzyme substrate complex complementary to the substrate. The active site binds with the substrate, forming a enzyme substrate complex. EX is converted into enzyme products, which subsequently dissociated into enzyme and products. How enzyme activity is expressed. Mention the difference between Keitel and IU expressing. Units of enzyme activity. Enzyme is usually present in biological systems in amounts too small to measure quantitatively. This is why enzymes are never expressed to terms of their concentration, but expressed by their activities. Various methods have been introduced to estimate the enzyme activation. Factors affecting the enzyme activity. Factors are like substrate concentration. The rate of enzyme catalyzed reaction increases with increasing substrate concentration until a maximum velocity Vmax is reached, and then there is no further rise of velocity in spite of further increase in substrate concentration. Temperature. The velocity of the reaction increases with the rise of temperature, reaches a maximum value at optimum temperature. After further rise of temperature, the velocity decreases due to the loss of enzyme activity by denaturation. Optimum temperature 40 to 50 degree for animal enzyme and 56 degree for plant enzymes. pH. Each enzyme has an optimum pH at which the velocity of enzyme activity is maximized. Below and above this pH, the enzyme activity is much lower. At an extreme pH, an enzyme becomes totally inactive. Most of the enzymes of higher organisms show optimum activity around pH. Enzyme concentration. Each substrate are available, velocity of a reaction will increase with the reaction of coenzyme. Product concentration. Accumulation of products generally decrease enzyme activity. Products bind with active site of enzyme and form a loose complex. Thus, enzymes cannot bind with substrate. As a result, inhibit the enzyme activity. Enzyme inhibitors. Inhibitor decrease the rate of enzyme catalyzed reaction. Activators and coenzymes. Coenzymes and activators and coenzymes necessary for activation of certain enzymes. Time. Under ideal and optimal conditions, time required for coenzyme catalyzed reaction is under non-optimal condition. Enzymes will need more time. Light and radiation. UV ray, X-ray, gamma ray inactivation enzyme may be formed by peroxidase. For example, X-ray inhibitors, salivary and amylases enzyme. What is Km value? Km is the Michaelis constant. Michaelis constant is characterized of an enzyme and this particular substrate and reflects the affinity of the enzyme for the substrate. The Michaelis constant is a substrate concentration at which initial velocity of an enzyme catalyzed reaction is half the maximum velocity attainable at the particular concentration enzyme. Significance. A small Km reflects a high affinity of the enzyme for substrate. Large Km reflects a low affinity for the enzyme substance. A small low Km reflects a high affinity for the enzyme for substance. Large Km reflects a low affinity for the enzyme for substance. What is Q10? Q10 or temperature coefficient is a factor by which rate of biochemical progress increases in temperature. Regulation of enzyme activity. Regulation of enzyme activity in the living system temperature of pH is maintained with the relatively narrow range of products, usually do not accumulative, so different mechanisms are adopted by the living system to regulate the enzyme activity in a cellular level. Enzymes can be regulated by changing the quantity of enzyme, altering its catalytic capacity. Enzyme inhibition, the process by which enzyme inhibition diminishes the velocity of an enzyme catalyzed reaction is called enzyme inhibition. Enzyme inhibitor, any substance that diminishes the velocity of the enzyme catalyzed reaction is called enzyme inhibitor. Classification: enzyme inhibition, reversible, irreversible, allosteric. Reversible, irreversible, reversible, competitive and non-competitive. Irreversible inhibition, the reversible inhibition binds with enzyme by non-covalent bonds.